Really, I'd like to thank all the attendees for being here. Really, thank you for caring so deeply about variant annotation, particularly in the late July. Really, I mean, I think it shows your dedication to biomedical data science. And so, welcome to our first Data Scientist Roundtable. So, in the Data Scientist Roundtable, what we're going to do is ask 5 questions, each of 2 data scientists, in this case, folks that work on variant annotation platforms. And I think that's going to be very exciting today. I've been working with a lot of variant calling, variant annotation lately, as I'll show you in a second. And really, I feel really motivated. In fact, the last call I was on, there was a lot of mention of variant annotation. And so I think this is a really timely topic, really, as we have huge data sets and we really try to turn those into knowledge. So I'm Ben Busby. And if you're interested in other initiatives like the Data Science Roundtable, find me on LinkedIn or Twitter. I also run some hackathons. Those are fun to check out. So logistically, if you have questions during this webinar, please put them in chat or Q&A. Frankly, I see the chat a little bit faster, so if you have a pressing question, put it into chat. I'd also like to thank my colleagues Brenton and Caroline for running the logistics of this webinar, and they'll be helping out with the questions and so on and so forth as well. That'll be good. Again, the structure of this webinar, I'm going to ask both of our data scientists here 5 questions over about the course of 10 minutes, and they will probably enlighten us a lot about variant annotation. I'm going to give a brief overview of sort of variant calling and sort of setting up for this, why you would want to annotate variants, that kind of thing. And then we'll go ahead straight into the interviews. Hopefully everybody's ready. All right, let's go ahead and go. So basically, as I think all of the participants here know, the vast majority of you guys the vast majority of you are professional bioinformaticians. And so really here, I think, you're very familiar with these. Now we have massive data sets all over the place, not only DNA, but also RNA we can call variants from. So essentially we map short reads, or in this case, long reads. Shout out to PacBio and Nanopore for really making variant calling a thing. We map them to reference genomes. By the way, please switch to GRCH38 if you haven't. That's a personal request, not a professional request. But anyway, so we do that, and then we call variants. GATK 4 is definitely the most used caller, although we're seeing other callers come up, particularly for somatic low frequency type of things. I think all of us learned a lot about COVID in the past year, and so we saw sort of the emergence of low freq and other things with that as well. And then now that we have these massive populations, such as the UK Biobank, we can easily run GWAS on things like type 2 diabetes, and we see suprathreshold SNPs that we obviously correlate with some of these things. These might be in HLA, for example, or near HLA types, although obviously a lot of them are intergenic. And intronic, we do see SNPs in coding regions, particularly in rare disease, and complex combinations of them in more complicated diseases. And so these are the things we want to be able to dig into. Really, yeah, I mean, the DNA Nexus platform historically, although now we do a lot of genotype, phenotype, and imaging integration, has really been pretty oriented towards secondary analysis of genomic data. And so we can see all kinds of ways to view that with locus zoom and so on. And then, with these variant annotations and other things, really it's nice to be able to find these variants. And then the next thing, obviously, we want to do is take individual variants and really annotate them to be able to extract knowledge. Now, there's an organization called KG, and really KG has benchmarking challenges every year, almost every year, for various annotators, and that's great. But I remember something that Stephen Brenner, who sort of runs KG, said one time when we were in a meeting, in an online meeting before they were cool, actually, and that was I got nothing from the participants. But anyway, so and I remember he said, really, the biggest strength we see in annotations really are when we have some clinical evidence, some structural predictions, as well as some population evidence. And so really that has pointed me to really thinking a lot about variant annotation, not in terms of individual annotators, but platforms which can integrate a lot of information. And so today, I'm really excited because we have two such platforms, one which is very modular and folks can mix and match a lot of information, and one which is sort of a really sort of solid, complete package. I think you guys will be able to see that as well, and that'll be very exciting. So without further ado, I'd like to introduce Kim and Stefan, who will be demonstrating JHU's OpenCRAVAT and Bio-Product's Helix Labs variant annotation platforms. And furthermore, I'm particularly excited about these folks because really I think they speak to sort of different use cases, as you'll see, in variant annotation. And so these two platforms, I think, actually complement each other relatively well, rather than being sort of in direct opposition to each other. And so that'll be cool. But like I said, the format of this is that I will interview each of them for about 10 minutes, and then we'll bring everybody back together for a panel discussion on variant annotation. So for those of you who are watching the clock, I think it'll be about the rest of this webinar will be about 25 minutes or so. And also, on a personal note, I would love to hear feedback about this format, whether you liked it, things to improve, and so on. So please feel free to reach out to me on LinkedIn or Twitter, and happy to talk about it. So without further ado, I think we're going to start with Kim. Kim, are you ready for for 5 questions about variant annotation?
Absolutely. Let's get to it.
Awesome. All right. So, Kim, in your view, why is variant annotation important? I mean, in other words, why have you devoted years of your life to working on a variant annotation platform?
It's a formidable question. But the truth is, as sequencing is getting cheaper and cheaper and cheaper, we're being buried in an absolute avalanche of genetic variants. And we've got this task to understand and find the ones that are important to human health in some way. So in this setting, what's key is the concept of prioritization. By using variant annotation tools, even just a few simple things like allele frequency, we can take something like 3 million variants in the human genome and whittle that down, list down to a handful, maybe a few dozen of variants that might be relevant to that person's health. Then, using variant annotation tools, we can really get in depth about those variants, learn about them, what they do, what they've done in the past, all the background information that exists. The philosophy of OpenCRAVAT is somewhat unique in the field of variant annotation software. We think it's important to find and integrate new information quickly so that we can make the analysis as custom as possible to what the research problem is.
Awesome. Well, that was a great answer, and you are ahead of the clock, but I am hoping to ask some follow-up questions about what you might do with ranked variants once you have them. But let's save those towards till the end. And go on to question number 2, which is, how does your platform interface with end users, say clinicians and biologists?
Okay, this is really interesting because as I've worked face-to-face with the users of annotation software, I've realized that there are 2 groups of folks who are using the software. One group of folks is clinicians and biologists. They have a need to have a tool that is not too complicated and maybe doesn't include a command line interface at all, because there is a significant startup cost to using those kinds of tools, installing them and configuring them. So from that end, We've made the software really easy to install. You never have to touch a command line. You just double click to install and pull up the interface. Then when someone's doing an annotation, there's an app store available of different resources that may or may not be relevant to the analysis. And clinicians and biologists have a pretty good handle on most of what they're interested in already, so they can select those using simple checkboxes, but also use the App Store interface to learn about new resources or things that are slightly outside their scope of knowledge, so they can incorporate those as well without doing a ton of extra background reading.
Fantastic. Thanks. And I want to now flip that question around and say, how does your platform interface with folks we might call power users? In other words, computational biologists.
Power users are the best because they really bring up the interesting questions and how to handle the data. So we don't want to constrain them within the graphical interface that clinicians and biologists can use. There's a back end to OpenCRAVAT that can be operated solely from the command line. It can be installed with a single pip command, so it's not a problem for them to get it. And it's also not a problem for them to run it. Power users can build annotation commands by simply tacking on extra options to best suit the data that they've got. In the case of power users with many variants that they'd like to analyze, we have some preliminary analyses of about 5,000 or more samples, and we found that OpenCRAVAT does work very effectively for large-scale data. While we're not there yet for millions of samples, it is possible pretty easily within OpenCRAVAT to tackle the data in smaller chunks and then merge it all together.
Awesome. Okay, and we can in a different webinar we can talk about merging millions of VCFs and what that would look like. But that's a whole different topic. All right, what sets your platform apart from other platforms out there? There's a lot of great variant annotation platforms, and we weren't able to interview everybody from all the variant annotation platforms.
There are a handful of very popular variant annotation platforms that we are kind of competing with, and they all are great and have strengths that cannot be minimized. We try to take those strengths and steal what we can and innovate beyond that. So I'd like to think that we do what most of the other tools do, a little bit faster, and with the main added bonus of portability to different systems on a laptop, on the cloud, on clusters, and the ability to customize analysis. So users can go to the App Store and pick out, for example, only cancer-related annotations and really narrow down their research to cancer-relevant annotations. Or they can do so for cardiovascular disease or for non-coding variants and regulatory regions. By considering these options upfront, users have more control over the annotation that they're doing, the depth of the analysis that they're doing. And since the App Store is open, I haven't really had a chance to discuss this. Users can submit things to the App Store themselves. Resources that aren't available can be added with a little bit of coding and a GitHub upload command, basically. So that really makes us stand out from other existing platforms. The mix of an app store that you can contribute things to yourself, or you can add things to and just not publish, and the MIT open source license means that it's really a playing ground for companies or institutions or groups to take the software and completely make it your own. Fantastic.
Thanks for that. And finally, why do you collaborate with DNA Nexus? What is it about DNA Nexus that is compelling to you?
Well, DNA Nexus is the leading cloud-based genome science platform, and it solves a challenge that I've personally encountered, where it can be really difficult to get software running on cloud platforms in general. It's kind of clunky logging onto the system, getting your code running there. But DNA Nexus has an app architecture and that drag and drop interface that you showed in an earlier slide. That makes it really easy for folks, even without computational expertise, to import OpenCRAVAT into their project, drag and drop the files that are the inputs and outputs, and they can scale it up immensely without any command line knowledge being necessary.
Awesome. Well, thanks for answering all those questions. And actually, you have a few minutes left, and so I think what I'd like to do is ask a couple of follow-up questions, and then if there are follow-up questions from the audience, Kim has another 3 minutes allotted to her. So the first question, really sort of a burning question I've had, is how many variant annotators are there on the OpenCRAVAT platform right now?
Well, it depends how you count them, because there are some tools that have sub options. For example, the CHASM plus mutation prediction algorithm predicts if missense variants, somatic missense variants in cancer are more or less likely to be driver variants. There are multiple alternative versions of that are specific to cancer subtypes, in total a few dozen. So if we count them all collectively, the number of annotation apps is over 150.
Wow, that's a lot. So a question from the audience that, can you give some other examples of annotation apps that are on the platform?
Okay, well, there's the kind of basic things people want, like allele frequency databases. There's NOMAD2, NOMAD3, 1000 Genomes Project, and a few older allele frequency databases if they're still needed. A wide variety of in silico pathogenicity prediction methods, both for missense variants. There's one for indel variants. We're trying to expand into that field, but it's a little complex logically. Splice variants, non-coding variants, so that we've placed a real emphasis on those because myself and the lab head, Dr. Karchin, have a lot of experience in building in silico predictors, and we think it's very valuable to provide them all easily in a single setting, as it's virtually impossible to download my code, at least, and run it from scratch of the tools I've developed. So if someone had to do that for dozens at a time, it would be intractable.
Wow. So then let me ask one last question. And I'll probably ask the same final question to Stefan, which is, then say you've ranked variants. Can you talk about some use cases that for sort of optimizing ranked variants that OpenCRAVAT is really specifically good at? Noting, say, for example, the splice stuff you were talking about just a minute.
Yes, I can bring up two examples offhand. We were funded by the ITCR, which is a cancer-based group. So we're very great at finding and prioritizing somatic variants in cancer. There are several databases for known driver variants and driver regions and hotspots, as well as several predictive methods. And those can be used in combination with things like spice predictors to find variants that, while not previously known, are pretty suspicious in terms of their relevance within somatic variant, progression of cancer. Another example is not somatic variants in cancer. You brought up the KG project. There's a challenge ongoing right now where folks who have submitted their DNA to the Rare Genomes Project are seeking to have the molecular cause of their rare disease ascertained. And they have a set of 30 or so families where the goal is to rank the variants in that genome and see if you can find the ones causing the rare disease. OpenCRAVAT has been found to be, on our initial test, incredibly effective at identifying those variants by incorporating gene level information, pedigree information because they're family level data, predictors, genome constraint, and allele frequencies. With really only like 5 or 6 pieces of information, we've got great accuracy on pinpointing those variants. It really is a playground.
Fantastic. Well, I want to say thank you very much, Kim. And now we're going to turn the floor over. We're going to rewind the slides and turn the floor over to Stefan, who is going to enlighten us about his variant annotation platform. So without further ado, Stefan, to your mind, Why is variant annotation important?
Formidable question, Ben. So at Bio-Prodict, we've been building these systems for protein analysis for over 10 years. And in our search for new data and incorporating new solutions, we found that there are lots and lots of data sources that are available for variant annotation. And that's because, well, there's a lot of call for it. It's very widely applicable. Many different fields like pharmaceutical field, the clinical fields, they're all looking for ways to annotate their variants and be able to analyze them better. So that's why at Bio-Prodict we made our product, which is Helix, and we were inspired to add novel contributions to variant annotation. We don't just build stuff that's based on data that's out there, but we actually build novel stuff based on our own systems. And being able to extend the coverage of data that's in like ClinVar and Nomad, that's of enormous value to us and to enormous value for users. So that's what inspired us.
Awesome. All right. Well, let's move right along to question 2, so we have time for bonus questions at the end. All right. And so let's talk about the same kinds of end users we talked about with Kim. How does your platform interface with biologists and clinicians?
Exactly. So if you're a clinician, if you're a biologist, if you're an end user, basically, that means that we have this amazing platform available online. You don't have to install anything, and it delivers very high quality reports on variants. Any variant in the human exome, and we also do all other organisms, it's available on our platform. You can just type it in, use a transcript identifier, and you get a variant report that includes our pathogenicity predictions that are completely novel, that don't incorporate external predictors. You get references to literature that includes not also the exact variants that you're looking at, but also variants that are inside homologs or even in different organisms. And you also get insight into evolutionary pressures because we build these systems on very, very deep and very detailed multiple sequence alignments. And you get all that data in a very nice, concise, well-defined, well-designed even report that you can even download as a PDF.
Awesome. All right, cool. Moving right along, and I will warn you, I'm going to ask questions about homology at the end, because I think that's really cool. All right, so then let's move along. How does your platform interface with power users?
So power users, they're awesome, and we're power users ourselves for many different types of software. So we based our interaction with them on our own experience. So what we did is, basically the first thing we did is build an amazing API. We use Rust, which you might be familiar with, to deliver a very stable, very fast, responsive API that our power users can interact with. We also have large, well-documented large API documentation with examples and the code you can expect to use. And we really hammer home the point that you can hammer our API. If you're a power user, if you want to shove 10,000 VCFs through our systems, that's completely fine. And we're going to make the experience the best for you that we are able to. Now, we're also working with custom tooling that's still in development, but we want to be able to process VCFs for you and integrate that, of course, with DNA Nexus, because, well, power users are sometimes also DNA Nexus users. And finally, because if you're a power user, you have very specific needs. We prefer a collaborative approach. So we sit together with you and we see if we can adjust our experience to your needs. And that's one of the major advantages of Bio-Prodict schemes.
Fantastic. I think you've touched on this a bit already, but to ask the question explicitly, what sets Bio-Prodict Helix apart from other platforms?
So Bio-Prodict Helix is a commercial product. We are not a charity, we're not a university. That's completely fine. There's a space for that. So if you want access to our full proteome predictions and annotations, it's not going to be free. We do have some predictions that are free, but the bulk of it isn't. Now, that's a drawback, but of course there are some benefits to that. That means that you get updates to our entire database twice a year, at least. So whenever there's a new version of ClinVar, whenever there's a new version of Nomad, we incorporate those into our technologies and we try to make them better. So constant updates, constant work. We're funded for the long term, and this is not just a postdoc project that might expire in like a year. Second, you get access to all these hosted reports. We host them directly. You get access to them in like 10 seconds, and you don't have to write your own software. And finally, just as I said, we offer this professional support. So if you want to contact us, just send me an email. I will talk to you directly, and we try to adjust our algorithms, our experience to your needs. And that's something that, well, we can offer as a commercial platform.
Fantastic. Last stock question. Why DNA Nexus? You touched on it, but why are you really interested in working with DNA Nexus clients?
Right. So to me, as a developer, DNA Nexus is basically like a match made in heaven. So I like to focus on the data scientist, focus on gathering data, getting the best experience for our users out there. But doing interfaces is really, really hard, and making these pipeline products that are robust and work well is really, really hard. And being able to offer that through a partner like DNA Nexus, that's just amazing to me, because we can build these products, we can focus on what we do best, doing the data science, doing the work to get the best data out there, and then our users can integrate it into their own workflows for basically zero cost, the extra cost that is. So that's why we love to work, and we love to work with DNA Nexus and get the best experience for our users.
Awesome. All right. So I think I have two follow-up questions, and I promised you I was going to ask both of them actually. The first one is homology. Can you explain to us quickly why homology makes variant prediction stronger?
So we find that homology is very important for variant prediction, because there are a lot of proteins out there that are really similar to the protein that people are researching, but it's difficult to access them, right? So what we do is we have these very big, very detailed multiple sequence alignments, and that means that we can take a look and see, hey, this specific protein looks a lot like this other different protein that exists in an entirely different organism or is described as an alternative protein, for example, in the human body. So a great example of this is HDAC8, which is important for human health, but there's a homolog there that's called HDAC1. So if you're researching HDAC8, a large part of HDAC8 it also occurs in HDAC1. There's different literature for that, there's different predictions for that, different data. But if you don't know about it, you might never get access to all of that. Whereas with Helix, you get access to that data because we can make that connection, even to homologs that you might have never expected. I mean, HDAC8, HDAC1, you're gonna guess. But if it's in a horse, who knows what data is out there? Who knows what literature is out there? Maybe there's like a free amino acid insert. You'll never be able to find literature for that by searching for the exact residue you're looking at. But with Helix, that's all gathered in that single report, and that offers so much more information than you would get regularly.
Fantastic. And so then I think that leads into my last question to bring it all together. What use cases, what specific use cases does your platform enable that maybe some of the other variant annotation platforms out there don't?
So very specific use cases that set us apart. Well, of course, we do everything that, for example, Revel or CAD can do. So that's not really unique there. But what we can offer is full exome coverage. Every single variant is covered, and not just in humans, but we're also working with different organisms. And that's where the power comes in, because that means that we can explore this entirely new world where we can, for example, look at protein engineering. We can look at organisms like bacteria or yeasts, for example. So these are entirely new ways of looking at this data and incorporating data from different sources that Helix can offer that other projects might not.
Well, thank you. That was really, really fascinating, and I would like to say I greatly appreciate both of you being here today and really sort of giving us an idea of the landscape of variant annotation platforms. And so I would like to encourage everyone on the call, if you're working with DNA Nexus or you're thinking about working with DNA Nexus, contact your sales folks or whoever, or if you're a UKB wrap user, reach out to the community about variant annotation, and we'll get you in touch with Stefan and Kim, and that's fantastic. And really, I'm very excited, really about the future of genomics and really getting genomics into the clinic, into the EMRs. And I think these platforms, in my opinion, give us two examples where we can really extend research a lot in terms of correlation with variants in disease, like we might work with something like UK Biobank, but then also getting decision support to clinicians thinking about not only variants, but combination of variants and how they affect cellular networks. So really, I hope you've enjoyed. This is, again, the first data science roundtable we've done on DNA Nexus. I think during the talk, we've really been able to address questions from the community, those of you out there. And I thank you for attending once more. And thank you to Stefan and Kim for being here. I hope you have a great rest of the summer. And we will have another one of another data science roundtable in September, most likely talking about workflow languages. So thanks again to Stefan and Kim. And we will hope to see you soon. Thanks, everybody.